My name is Juha Hulmi, and I am professor in exercise physiology here at the Faculty of Sport and Health Sciences at the University of Jyväskylä. My research group aims to understand metabolism and physiology of exercise and muscle using a variety of in vivo and in vitro research models. Some of the effects of resistance training, such as the number of muscle nuclei and DNA methylations, can persist in muscles for weeks even after a training break, which may explain why muscle size gains can be rather fast when training is started again after the break. The question is, does resistance training leave protein memory traces as well? In the present study, 10 weeks of resistance training was followed by a break of the same length and then followed by another 10 weeks of resistance training. Another group served as controls. Using high-end mass spec proteomics, we studied the quantities of over 3,000 muscle proteins. We first found that resistance training affects on muscle proteome and that the effect is reproducible within individuals. Next, we found two main types of change profiles in muscle proteins. Some proteins changed as a result of training, returned to their pre-training state during the break, and changed again during the second training period. These so-called reversible proteins included proteins related to aerobic metabolism. Another group of proteins changed as a result of training and remained changed during the break and after the second training period. Among these so-called retained proteins were, for instance, CBR1 and calcium-binding proteins such as calpain-2, whose gene has recently been identified to retain an epigenetic memory trace after a training break. Therefore, even though muscles eventually shrink back to their original size during a rather long resistance training break, a proteomic memory trace of previous training remains in the muscles. This may mean that we don't need to start from scratch when we get back to lifting. Thank you for your time to learn more about our study published in the Journal of Physiology. The full paper was published in volume 603, issue 9. Please contact juha.hulmi@u.fi if you have any questions.